everybody, welcome. So today we are going to be looking at membrane transport demonstration. So I am blocking the way here, so I'm gonna move myself. But dialysis tubing is basically this plastic looking tube that is actually made out of cellulose. So if we look at dialysis tubes structure, we will see that it's literally just a bunch of fibers of cellulose. I don't think I'll be able to find it. Maybe if I had cellulose here. Oh well, it's not cooperating me. But basically just imagine a bunch of strips of this cellulose, right? And they're all like overlaid over each other in order to form this sort of sheath of cellulose, right? Now, you'll notice that instead of just drawing it like this, where it's completely opaque, I showed like these cellulose fibers overlapping each other, and you'll notice there's a lot of empty space here, right? So imagine these as pores, and these are just microscopic pores, R-E-S, and it allows for particles to go through them. But large particles can't go through them because, well, something tiny, teeny tiny, like this can easily pass through, right? It goes straight through. A bigger molecule that's this big, it can't go through it. So it's a little permeable. It is selectively permeable and doesn't allow for different size molecules to enter. It only cares about the size. The shape can affect it, but it's mostly the size. How big is the substance, the particle, the molecule? So, if the tubing is tied on one end and then filled with a solute solution, and then tied on the top end, you can create a cell model which is semi-permeable. If you place that cell into a solution, you can determine what types of substances are permeable through that membrane by testing for the presence of various substances using chemical reagents. What does this mean? So, we basically have this tube. We're going to tie it on one end, right here, see? A little tie. And then we are going to fill it with a solute. We're going to fill it with something. And then we tie it at the top end. And this is a semi-permeable membrane, as we just said, right? Cellulose is semi-permeable. And if we place this make-believe cell, so we pretend that this dialysis tube is a cell, we can then figure out which type of substance is permeable through the membrane by testing for the presence of various substances using chemical reagents. So we can just test to see which substances are permeable, what can go out, what can go in. Now, we have the drawing here, so we're gonna go through that. Zooming in. Okay. So at the start of dialysis, we have our solvent. We're gonna think of this as our, I believe, Benedict solution, but I could be wrong. But what we're looking at here more is just how these solutes distribute themselves. So we notice these are blue, right? These are red. Blue is bigger than red. So blue just stays inside of the dialysis tube. It does not leave, but the red does go out until it is at equilibrium and has distributed itself in equal amounts across the dialysis tube and the solvent itself. At the beginning, there's a concentrated solution, but now it's a diluted solution, or at equilibrium. And what we're going to do in this demonstration is we basically just had a starch solution, which was whitish in here, and then we had a bunch of iodine surrounding it, and starch is white, right? So we have this starch right here. Wait. So it's not cooperating with me, but starch is right here. And it's whitish. And I2, think of it like this. I2 is just two molecules covalently bonded to each other. But starch, let me just scare you with a picture of it. That's not a scary picture. Oh, come on. They're being generous. One. Well, starch is real big. See, this is like a simple starch, I guess. We already have two atoms here, and then two atoms here, and overall, it's so many atoms. Oh my god. Compared to this thing, it is huge, so it cannot go through it because remember those pores? The starch is way too big for those pores, but the I2 isn't, so it goes into the dialysis tube. And what this does is it causes a reaction, right? When they meet, the mixture turns bluish black. What has happened? Iodine enters the cell and reacts with the starch, making it turn black, and the starch does not exit the cell because of its large size. Alright, now we have a thinking question. Suppose that you placed starch and glucose into the dialysis tubing cell model, and then you did the experiment. So we're putting starch as well as glucose. So starch is a big molecule, glucose, remember, is just two smaller molecules with a glycosidic bond. It is, oh my god, I just learned this. Glucose is, oh, it's just one thing. Okay, sorry. I was mixing it up with maltose and like galactose and lactose. Those are what I was mixing up with. But anyways, starch is much bigger than glucose. And then you tested the sampled solution for the presence of glucose using a special reagent known as Benedict solution. So here we have iodine, which turns blackish blue when it interacts with starch, and we also have Benedict solution, which changes from blue to a reddish color if monosaccharides and glucose are present. So, glucose is small enough that it can easily, let's bring back our little pores here. So, this is a glucose, this is a glucose. They can easily go through it. Now, something like starch, pretend this is the inside of the cell, that's the outside. Starch is way too big, so it cannot go through. It stays in this spot. But remember, iodine, it can go through. So but glucose is small enough that it can actually leave it. And since the Benedict solution is outside, it will react with the monosaccharide glucose. So we'll see the inside dialysis turn black because the iodine entered and made the cell black, but we will also see that the solution surrounding the cell turns orange, and that means that glucose is permeable and can pass through since the glucose or the monosaccharide reacts with Benedict solution. The change in color shows that the glucose can leave and that it is permeable to glucose. The cell membrane is permeable to glucose. Anyways, that's it. Thank you for joining us. Have a lovely day, everybody. Take care.